Assalamualaikum warahmatullahi wabarakatuh. Alhamdulillah, today we have reached to the third subtopic of redox equilibrium, which is the voltaic cell. One example of voltaic cells in our daily life usage is the battery. What is actually a voltaic cell? Chemists also call voltaic cell as simple cells. Simple cells consist of two different metals which are dipped into an electrolyte and connected with the connecting wires. Simple cells are also known as voltaic cells or galvanic cells. These cells convert chemical energy to electrical energy. And these cells also is one of the example of redox reaction. Redox reaction causes the flow of electrons. Potential difference in this cell will be detected by voltmeter that indicates the presence of electric current. There are three types of voltaic cell, which is the first one is the simple chemical cell. The second one is the Daniell cell type 1, and the third one is the Daniell cell type 2. What are the differences between these three cells? For simple chemical cell, it consists two different metals, which is magnesium and copper plate, that are connected to the voltmeter by connecting wires. These two electrodes are immersed into the copper chloride solution. This cell uses a beaker as one of the apparatus. For the second voltaic cell, which is Daniell cell type 1, the cells consist of two beakers. The first beaker is filled with copper sulfate solution. Okay, the copper sulfate solution is inserted with copper electrode. The second beaker, which is actually a porous pot, in the porous pot, zinc sulfate solution are filled. The zinc sulfate solution is immersed with zinc electrode. Zinc electrode and copper electrode are also connected to the voltmeter by connecting wires. So in this Daniell cell type 1, we use only one big beaker filled with a porous pot. I will give you the function of porous pot later on. The next Daniell cell, which is Daniell cell type 2, consists of two beakers. The first beaker is filled with copper sulfate solution. There are copper electrodes immersed in the electrode. The second beaker consists of zinc sulfate solution. There are zinc electrodes immersed in the zinc sulfate solution. The two beakers are connected together by a salt bridge. Actually, the function of salt bridge and porous pot in Daniell cell type 1 is the same, which is to allow the flow of ions through it. Okay. Both these the three cells produce electrical energy when there are transfer of electrons in the electron. Now, let us discuss what is the chemical reactions occur in the simple chemical cell that can allow this chemical cell produce an electrical energy. In this cell, magnesium is more negative because the E0 value of magnesium is negative. So, magnesium has higher ability to become negative terminals. Magnesium will release or donates electrons to form magnesium ions. Then, the oxidation reactions occur in the magnesium ribbon. This is the half equation occur at the negative terminals. On the other hand, the E0 value of copper is more positive. That makes copper becomes the positive terminal. Copper ions from the solution, copper chloride solution, receive electrons to form copper atom. Therefore, the reduction reactions occur. The half equation occur at the positive terminal is copper ion receive electrons to form copper atom. Okay, now if we look at the electrons flow here, electrons will flow from the negative terminal, which is magnesium ribbon, towards the positive terminal, which is copper plate. Okay, we can see the arrow shows us the flow of electrons. Now, let us look at the animation for the reactions that occur in the simple cell that we have just discussed. Okay, this is the simple cell. When magnesium ribbon release the electrons, magnesium atoms will become magnesium ion. Bear in mind that ion, magnesium ion, will dissolve into the solution. When atom, which is a solid, form an ion that dissolve into the solution, it will make the magnesium ribbons dissolve into the solution. So we can observe that magnesium ribbons will become thinner. On the other hand, when magnesium atom release electrons, the electrons will flow from the negative terminal towards the positive terminal. I have mentioned before, at the positive terminal, copper ion from copper chloride solution, okay, bear in mind that copper ions comes from the solution, will receive electrons and becomes copper atom. Copper atom is in the form of solid. Therefore, at the end of the experiment, we will see a lot of copper atom deposited on the surface of copper plate. But what is the correct observation that we can observe? Now look at this animation. Okay, we can see that the magnesium ribbons becomes thinner or smaller, while at the positive terminals, okay, we can see the brown solid deposited on the surface of copper plate. It makes the copper plates look bigger. If the reactions occur for a longer time, we can see that the magnesium, as the magnesium becomes thinner and smaller, the copper plate also becomes bigger because there are brown solid deposited on the surface of copper plate. Okay, this is the half equation occur at the negative terminals, where I said that the magnesium atom, which is in the form of solid, release electrons to form magnesium ion that dissolve into the solution. Okay. Magnesium strip becomes thinner or smaller. This is the correct way to write the observation at the negative terminal. While at the positive terminal, copper ion from the solution will receive electrons to form copper atom. So copper atom, metal atom usually exists as solid. But in order to write a correct observation, we must include the color of the solid form. That's why the observation is written as brown solid deposited. If the question asked you about the color of the solution, we know that copper ion is blue in color. Okay. If there are presence of copper 2 ion in the solution, the color is blue. As the copper ion receive electrons, that means the quantity of copper ion in the solution decreases. When the quantity of copper ion decreases, we say it as the concentration of copper ion decreases. Therefore, the blue color will become paler, or intensity of blue solution decreases. The correct words is intensity of blue solution. Okay. Now, does the reaction in the simple cell equals or same with the Daniell cell? So let us look at the apparatus set up in the Daniell cell. Daniell cell has two types of cells. Okay, the first one using one beaker and a porous pot, like this one. The second one using two beakers connected with salt bridge. As we see, the type of electrode is still the same, which is zinc electrode and copper electrode. So basically, the theory is still the same. Zinc will become negative terminals, okay, while copper will becomes positive terminal. Okay, it has written here. Okay, no need to write it again. So basically, the process is the same for both simple cells and Daniell cells. Okay, let us look at the theory. So bear in mind, when we use Daniell cell, it is a chemical cell, still a chemical cell, that also be constructed by combining two half cells with different E0 values. The Daniell cell is a voltaic cell where zinc and copper metals are used as electrodes, which are then dipped into their respective ionic salt solution. So if we can see, the copper electrodes are immersed into the copper sulfate solution, while the zinc electrodes are immersed into the zinc sulfate solution. That is the first rules of Daniell cell. Both salt solutions are connected by a salt bridge or separated by a porous port. Now, let us look at the E0 value of each electrode. So E0 value for two half cells are zinc is negative 0.76, okay, which is more negative. When the E0 value is more negative, it will becomes a negative terminals. But for copper, the E0 value is more positive. Metal with the E0 value that is more positive will becomes positive terminal. Okay, so bear in mind that the negative terminals where oxidation occur, okay, and the releasing of electrons occur, it is called as anode. Conversely, the electrode where receive of electrons occur, terima electrons, okay, terima electron, where terima started with letter T, so it's called as cathode. Cathode terima, cathode terima electron. Okay, so this electrodes is where the reduction reaction occur. So now we have another terminology. The electrode with the E0 value, which is more negative, will become negative terminal. Negative terminal is also known as anode because at the negative terminal there are releasing of electrons, while metal with the more positive E0 value will become the positive terminals. At the positive terminal, there are process of receiving electrons. Okay, receiving electrons, the electrode is called as anode. When the process undergoes is receiving electron, that means the reduction reactions occur at the electrode. Now let us see what is actually happens in the Daniell cell. Okay, the E0 value of zinc is more negative. We have mentioned this before. zinc will become negative terminals, which is anode, because zinc atom will donate electrons to form zinc ions. Therefore, oxidation reaction occur. The half equation at the negative terminal is as mentioned in the slide, where zinc atom release electrons to form zinc ion. Okay, while copper, the E0 value is more positive. Therefore, copper will become the positive terminals. We call this terminal as cathode. Why cathode? Because the copper ions from the solution receive electron at this electrode to form copper atom. The reduction reaction occur and the half equation occur at this electrode is copper ion receive 2 electrons to form copper atom. Every time when we see the half equation, okay, we must bear in mind that after all, we have to write the full ionic equation. Same with the process in Daniell cells or voltaic cells or simple chemical cells. Now, let us remind and memorize again how do we write the full ionic equation. We need to have the half equation at anode, okay, for the oxidation reaction. Next, we also need the half equation that occur at the cathode, which is for reduction reaction. Make sure that the number of electrons are the same. 2 electrons, 2 electrons, 1 electrons after the arrow, and the next one must be before the arrows. Okay, if we found the electron is the same quantity, then we cancel out the electron. Then combine both half equation together. So we will get the overall ionic equation as zinc atom added with copper ion to produce zinc ion and copper atom. Now, in this slide, we can see that the voltaic cell can be written in the form of cell notation. What is cell notation? Okay, this type of writing is called as cell notation. Okay, anode is written on the left. This is for the anode. Okay, and the cathode is written on the right side of the cell notation. This is the reactant and product of the cathode. So this is the reactant of the anode, while zinc ion is the product. Okay, copper ion is the reactant at the cathode, but copper atom is the product. This writing method is separated by a single line here. Okay, to separate the reactant and product, and double line here to separate the anode and cathode. actually the second, the parallel line is a salt bridge or porous spot. Now let us discuss how to use this cell notation. So from this cell notation here, we can see that the electron involved is zinc and copper. Zinc is more negative, while copper is more, E naught value of copper is more positive. So regarding this E naught value, we can predict the voltage reading or potential difference when we use these two electrodes. So the formula that we are using to calculate the potential difference is E naught cells is equals to E naught cathode minus E naught value of anode. So in this case, E naught for this cell, okay, the whole cell, is E naught cathode, which is 0.34, here, positive 0.34, minus E naught value of anode, which is negative 0.76. So when negative, okay, negative bertemu dengan negatif, it will becomes positive. So 0.34 plus 0.76, we will get plus 1.10 volt. This is the voltage value of the cell. Let's say we are using a nickel as electrode and pair it with lead. So we see at the E naught value for both electrode. Okay, so let us discuss this together. The E naught value of nickel is negative 0.25. So it is more negative. So that's why nickel becomes the negative terminals, while the E naught value of lead is more positive. Even though the value is still negative, but it is nearer to the zero, so we call it as more positive if we compare to nickel. So because it is more positive, it will become positive terminals. Okay, this is T actually, terminals. Okay, so therefore, if we are using the same formula, E naught of the cell is equals to E naught value of cathode minus E naught value of anode. So E naught value of cathode is negative 0.13 minus E naught value of anode is negative 0.25. So negative 0.13 will be added with 0.25. Therefore, we will get a positive 0.12 volt. So the potential difference of cells when nickel and lead are paired together is 0.12. Okay, how about this second example when we are using platinum, okay, as one of the electrode, and also both are using platinum, but the ions or substance in the solution is different. Here we will use bromide ion and bromine molecule, chlorine molecule, and chloride ion. Okay, now because platinum is inert electrode, okay, the substance that will receive or release electrons is actually the ions in the beaker. Okay, so we have to look at the E0 value of both substances, which is E0 value of bromine, which is more negative. Okay, that's why bromine and bromide are situated at the negative terminals. Okay, while the E0 value of chlorine and chloride is more positive. Okay, that's why chlorine and chloride are situated at the positive terminals. Okay, how do you know where does this substance is situated, whether at negative terminal or positive terminal? Okay, remember at the left hand side it is for anode or negative terminal. At the right hand side it is a cathode or positive terminal. Okay, these both substances is separated by salt bridge or porous point. Okay, so we want to predict the E0 value of the cell. E0 value of cathode is plus 1.36 minus E0 value of anode is plus 1.07. Therefore, we minus it, simple mathematics, and we will get plus 0.29 volt, which is this is the potential difference of the cell when bromine and bromide are paired with chlorine and chloride as the electrolyte. Okay, so thank you very much. I hope we will get something from this discussion. Inshallah, I will give you some exercises in order to increase your understanding on the voltaic cells subtopic. Thank you very much. See you in the next subtopic.